Okay, we're on to the final part of this section. These are some great pictures. It's a good summary. It's showing transcription factors. So here is a double-stranded section of DNA, and it's showing a gene right there. RNA polymerase is an enzyme involved with the copying of DNA. In other words, making a strand of messenger RNA. See right here, well, you can see it's being split. And then see this coming off. It says messenger RNA transcript, and it's in the form of codons, the three sections of nucleic acids, or well, nitrogenous base, I should say. Here is the DNA section. See the T, the G, the C, and then right here there's an opposite complementary strand of messenger RNA being made. So opposite the A, there's a U. See, this is placing an A across from the T. All this is accomplished by enzymes, and it's going to peel off like that, and then it's going to get to the end of a gene, and that's where it will stop. All right, showed you that when okay, now this actually happens inside the nucleus of a cell. This whole transcription portion, that happens in the nucleus. When it moves out into the cell cytoplasm and attaches to a ribosome, and it's going to start being translated at that point. So it says a large ribosomal subunit. You can see where they're kind of separated. It doesn't actually pick up like this. It's just showing here is a transfer RNA molecule. See, there's the MET start codon. And it says initiator transfer RNA is going to start the process of translation. You're translating the genetic code. And then here, like there's MET, here's glycine. See how it's coming along and it's dropping onto here. And then what happens, these transfer RNA molecules, they will break off and go back and go get other amino acids, depending on what the genetic code is. So it's going to read, see here's AUG, which is the start codon, GGC, and then it's going to read, no, that's GGC, this is GCC. It will keep reading the genetic code and then going out and getting the appropriate amino acid. Okay, just like that. Just a little bit bigger picture. See, this is showing, see here, this one's peeling off. So methionine, glycine, alanine, it looks like. And then it just keeps going on as long as the genetic code continues until it comes to a stop codon is when it officially will stop. But there's one that looks like proline, phenylalanine, there's tryptophan. And then here it says a stop codon, UAG. And let's see if that's actually right. There it is, UAG, stop codons. At that point, the whole process will stop. Now, here is a summary, and then we'll get to a little bit of practice, and that'll be the end of this. So right here it says transcription. Like I said, this all takes place. Remember, this is the nucleus of a cell. There's the nuclear membrane or envelope. Here's the rough endoplasmic reticulum. And so basically, here is the strand of, it says messenger RNA transcript. Now, don't worry about introns and exons, Those are sections that get removed, but that'll be for another day. So here's your messenger RNA strand right there. You see it's moving out. Remember, this membrane is selectively permeable, allows certain things to cross and things can't. And so once it moves out here, it's going to attach to a ribosome, and then here comes the transfer RNA, and of course it's going to, the first thing it's going to grab is the MET start codon, and then it will start building a polypeptide. And like it says over here, it binds to the ribosome and is translated by transfer RNase, producing a polypeptide. There's the polypeptide, and then, like it says here, a modified functional protein. And that's the whole process. So you start in the nucleus, it moves out to a ribosome, and now you have a protein. If you're wondering what are proteins, those are enzymes. You know, sucrase, maltase, lactase. enzymes that help break down food, enzymes that power metabolism, all that stuff we covered with energy conversion, the cytochrome C oxidate, all that stuff that's part of the electron transport chain, Krebs cycle, alpha ketoglutarate, all those things are enzymes. Okay, so now for some practice. You are going to do this in your lab. If I can get I'm going to make this a little smaller, get the line up. There we go. So here is a strand of DNA, and then we're going to make an opposing strand of DNA, and then we're going to transcribe it here, and then depending on what that code is, we will translate it here. And we'll do that next.